Hello to all the attendees from within the United States, Canada, UK, Singapore, and other international partner countries. Welcome to this presentation on the development of rapid viability RT-PCR method for detection of infectious SARS-CoV-2. My collaborators in this research project were Dr. Stacy Kane, Dr. Maher El Sheikh, and Ms. Tanil Alfaro from Lawrence Livermore National Lab. The picture on this slide depicts potential modes of transmission of SARS-CoV-2 and thereby spread of COVID-19 in the environment. Here is a disclaimer for today's presentation. Upon declaration of COVID-19 as pandemic last year, the EPA Office of Research and Development expanded its research program to be prepared for environmental spread related response. The development of SARS-CoV-2 RV-RT-PCR method was one of those projects. The research objective was to develop a rapid and sensitive method to detect infectious SARS-CoV-2 for environmental sample analysis to help study and understand COVID-19 spread or SARS-CoV-2 transmission via surfaces, and also to facilitate timely environmental epidemiological studies. The research goal was to develop a proof of concept RV-RT-PCR method as soon as possible. So why SARS-CoV-2 RV-RT-PCR method? As many of you know, during the first few months of the COVID-19 pandemic, most of the studies on surface stability of the virus were done using RT-PCR method, which can just detect viral RNA without indication of the presence of viable or infectious virus. Even as of now, most of the very few studies which focused on detecting infectious SARS-CoV-2 from surfaces use the traditional cell culture based method, which takes several days to provide sample analysis results. Traditional cell culture based method is based on tissue culture infective dose or TCID50 analysis in multi-well plate format or traditional plates with a limited sample analysis throughput. Therefore, a rapid, sensitive, and high throughput analytical method was needed. So what is the SARS-CoV-2 RV-RT-PCR method? The rapid viability reverse transcriptase PCR method integrates cell culture-based enrichment of the infectious virus in a sample with virus-specific RT-PCR assay-based molecular analysis. Most importantly, the RT-PCR analysis is conducted before and after the cell culture virus post-infection incubation. The cycle threshold CT difference of delta CT between the RT-PCR analysis performed before and after cell culture virus post-infection incubations determines the presence or absence of infectious virus in a sample. The algorithm for results interpretation is explained in the graph. The blue line graph shows the RT-PCR response for time 0, that is before post-infection incubation of the virus, while red line graph shows the RT-PCR response for time final, that is post-infection incubation of the virus. The change in CT value between the T0 and TF RT-PCR analysis has to be equal to or greater than 6, which will indicate roughly a 2 log increase in the RNA. Only then the sample is called to be positive for presence of infectious virus. In this slide, a process flow for RV-RT-PCR is shown. Actually, when we started developing this method, we thought that we can use ViroE6 cells that can grow in suspension. However, unfortunately, we could not get those cells. That's why we use monolayer cell culture and we use ViroE6 cells. There are two 96-well plates used. One is T0 96-well plate, which is before post-infection incubation, and TF 96-well plate is post-infection incubation. On the right side, the other steps are shown. Basically, virions are recovered from swab samples in a buffer. The suspension is vortex mixed, and it is concentrated using centrifugal ultrafiltration device. The retentate then is filtered through 0.22 micron because we are talking about environmental samples here, and other microbes and spores present in the environmental samples can interfere with the cell culture. 100 microliters of the filter suspension is pipetted in a well of a T096 well plate and another 100 microliters is pipetted in a corresponding well on T96 well plate. Both the plates are then incubated at 37 degrees centigrade for 1 to 2 hours for virus infection of the host cells to take place. After the infection incubation, the wells in the plate are the medium with unattached virus is removed, and the wells in each plate are rinsed with fresh medium, and after that another aliquot of fresh medium is added in each well. After that, the T096 well plate is stored at minus 80 degrees centigrade because this is the before post-infection incubation plate. While the TF96 well plate is further incubated at 37 degrees centigrade for desired number of hours. This is the post-infection incubation. After this step, the cells are recovered from each well and RNA is extracted and RT-PCR is carried out. Please remember the algorithm that the delta CT value between the T0 and TF analysis has to be greater than or equal to 6 for a sample to be called positive for the presence of infectious SARS-CoV-2. So here are some of the major steps in the development of the RV-RT-PCR method. We developed the method using a 96 well plate format. We used Vero E6 as the SARS-CoV-2 host cell line and used 35,000 cells per well for RV-RT-PCR analysis. We first developed the method using SARS-CoV-2 in suspension and determined optimum infection incubation time of 2 hours and also determine optimum post-infection incubation time of 9 hours for high sensitivity of detection. We use CDC's N1-RT-PCR assay for method development. Many of you are aware of the fact that last year we could not get swabs for any R&D work because of the high demand for clinical sample collection and analysis. Therefore, we evaluated this 2 hours infection time and 9 hours post-infection incubation time first with the swab swatches. The swab swatches are made up of the same material as the actual swabs used for clinical sample collection. We also incorporated sample concentration step using the Decon Ultra 15 centrifugal filter device. Finally, we were lucky to get some swabs from a company called Sanijan in South Korea and evaluated the method performance using actually spike swabs using CDC's N1 and N2 RT-PCR assays. Here are some of the results with the spiked swab swatches. I apologize for a very busy slide. Additionally, the data involved here involved complex experimental conditions. Therefore, I would like to request you to please refer to our paper published in Journal of Virological Methods and also our final report, which is published online for all the experimental details. For the best interest of the time, let us please focus on important columns in this table. Column 1 presents the plaque forming units that were spiked onto the swab swatches. Column number 3 presents the plaque forming units that actually went for RV-RT-PCR analysis following all the sample processing steps. And the last column presents the average delta CT values for each spike level and their replicates. As can be seen that all the rows for each spike level except for this row for 67 PFU spike level showed delta CT of greater than 6. That means we could detect up to 67 spike level of the PFUs. So bottom line message is that a low number of SARS-CoV-2 virions could be detected from swab swatches using combination of 2-hour host cell infection and 9 hours of post-infection incubation, which is a pretty good sensitivity of detection. So feeling very confident, then we evaluated the method with actual spiked swabs. Again, this slide is very busy. So let us focus on important columns. The column 1 presents again the estimated PFUs that were spiked on each swab at different levels. Column number 4 presents average delta CT value for the N1 RT-PCR assay. In column number 5 presents how many replicates for each spike level came out to be positive. Similarly, column number 8 presents delta CT value for N2 RT-PCR assay, and last column presents how many replicates of N2 RT-PCR assay came out to be positive for each spike level. As can be seen, the lowest number of spike PFUs that could be detected by both N1 and N2 assays was 35. And for both N1 and N2 RT-PCR assays, 3 out of 3 replicates came out to be positive for 35 level spike. However, unfortunately, we could not detect 18 PFU that were spiked on the swabs, both by N1 and N2 RT-PCR assays. However, 35 is a pretty low number of virions. Therefore, we can say that a low number of virions could be detected by this method. And such a low number was detected in just 17 hours total time to results, which is pretty good compared to the traditional cell culture based method, which takes several days. This slide shows estimated timeline for swab sample analysis. Following all the sample processing steps that I described, a total time for 22 samples to be analyzed could be 19 to 22 hours. If the laboratory operates on multiple shifts and adequate equipment is available, additional batches of 22 samples can be analyzed every 4 to 8 hours, which shows the very high throughput of the RV-RT-PCR method compared to cell culture based method. Here is a rough comparison of throughput and time to results for both the methods. As is evidenced that RV-RT-PCR method uses 96 well plate format, and thereby it's a high throughput format compared to traditional cell culture based method that can use the also multi-well plate format, but they have to use large wells, and thereby only 3 to 4 samples per plate can be analyzed. Most importantly, the time to results by RV-RT-PCR is in hours compared to days taken by traditional cell culture based method. In summary and conclusions, the RV-RT-PCR method allows analysis of a batch of 12 samples in about 17 hours of total time to results compared to several days taken by traditional cell culture based method. The method enables detection of a very low number of infectious SARS-CoV-2 virions from swabs. The method uses 96 well plate format for high throughput sample analysis and thereby allows for a smaller footprint in the laboratories. The method includes ultrafiltration based sample cleanup and virion concentration step for a better sensitivity of detection from environmental samples. Here are some of the impacts of the RV-RT-PCR method. The method allows rapid sample analysis for a rapid response and recovery. The high throughput format can increase the lab capacity. The RV-RT-PCR method uses a smaller footprint for sample analysis and will generate significantly less BSL-3 level laboratory waste compared to the traditional cell culture based method. The method can be a model for other RNA viruses. It could also be useful in screening for and testing efficacy of antiviral drugs. The method could be useful in testing efficacy of neutralizing antibody-based vaccines. As I said before, we have published the method development work in the Journal of Biological Methods. The paper came out online last month. Finally, I would like to acknowledge the research project team that included my colleagues from EPA Office of Research and Development, EPA Office of Land in Emergency Management, CDC, and my collaborators at Lawrence Livermore National Lab in California. Thank you for your kind attention, and I'll be happy to take any questions that you might have.